this is where we finished off. I had kind of just given a quick overview of the immune system and had mentioned that we have kind of, you can break it into different parts, right? 3 lines of defense, or innate immunity and adaptive immunity and so on. And so that's what I'm going to talk about today, is adaptive immunity. I'm hoping to finish that today and actually start, or sorry, innate immunity. I'm hoping to finish that as topic 13-1 and actually start on topic 13-2. Trying to make up for just a little bit of lost time so that we're not rushed right at the very end of the semester. There's probably going to be a couple of things I will skip, and I will mention that I'm skipping this or that, which means that you can learn it if you want, and if you want to put it on the final exam, that's great. Some questions might be applicable, but I won't ask you about it directly, like the complement system, which I haven't talked about yet. I'm going to skip that. So innate immunity is non-specific and fast. It's something that you're born with, and usually it's kind of considered to be 2 categories. One, sometimes people say the skin is category A, and B is category that includes cellular and chemical defenses. Some people include others of those things in with the skin, because the skin does have a lot of its own chemicals, and there are some cellular defenses that are tightly woven in with the skin. So let's kind of talk about some of these things. The list will not be completely comprehensive. Like, you could talk about things like how your stomach is acidic, and that prevents certain organisms from getting through and inside you and so on. And there's a lot of other kind of minor mechanical things in different parts of your bodies, right? Like your nose hairs, right, are apparently there to help prevent dust from getting up your nose. I don't want to talk about nose hairs, but I do want to talk about the skin. So like I said, the skin is usually considered that first line of defense, and it's a barrier that's protecting most of your body. And there's quite a few defense mechanisms involved with the skin. Some of them are kind of outlined here. And like I said, I want to kind of talk about the skin in general. So there's a picture of the skin, and the skin is a few layers of cells, and the outermost layer is actually dry, keratinized, dead cells, and they kind of flake off on a regular basis. And that is actually a protective mechanism. If you imagine organisms trying to colonize that skin, and they are falling off with those dead skin cells. The other thing about skin is it's kind of flexible and springy. So you can imagine mild jabs, right? You know, poking a mild jab, it's hopefully not going to puncture it. If your skin was a lot more brittle, punctures are going to be a lot more likely. So it's relatively flexible, malleable, and protects against a lot of things. And has a lot of secretions in it as well, which is some of the things that I want to talk about today. So a whole bunch of skin secretions. One is salt. All right, salt comes with your sweat, and it's going to inhibit actually the growth of quite a number of organisms. A lot of organisms do not like to live in a salty environment. Some exceptions: Staphylococcus loves it. Candida, the yeast, loves it, which is why they make good commensal skin organisms, because they don't mind the salt. But it actually does prevent growth of a lot of other organisms. It literally dries things out. Your skin also produces this oily substance called sebum, which is a little bit acidic, and it actually inhibits quite a few organisms as well. Particularly certain pathogenic fungi don't like acidic environments in general. And so that's another thing that's found in your skin. So this is just surface stuff, right? Kind of making a relatively dry, salty, inhospitable environment. Doesn't prevent them from living there at least a short time, but from permanently colonizing, this prevents many of them from doing so. That's why many of your skin microbes are transient. You may have hundreds of species on your hands. Most of them are not going to survive there for days. It's going to be a short amount of time. We also have these other things that are getting a lot of attention in the research area now. These things called defensins. They are antimicrobial peptides, and we're actually finding these things everywhere. We're finding them in the skin. I was reading about somewhere in, I think it was Peru or Ecuador. There's one of these indigenous tribes that they're kind of Stone Age, meaning they're just living with bows and arrows and things like that. And one of the things they do with infections is they find a certain type of frog, and they gut it, and they apply the skin of that frog to the wound. And so some researchers were looking at this, and it had these things, these antimicrobial peptides. And in the last 10 years or so, there's been hundreds of these things discovered. Humans have some of them, and and have antimicrobial properties. So by peptides, usually we mean maybe 10 to 25 amino acids together. And here's a diagram showing how one of them works. It actually interacts with the membrane of bacteria and kind of forms these pores there. And like I said, lots of interest in these things in terms of how they're related to immunity and all that. We're still trying to understand that, but they're found in our skin and in our blood, and they have antibacterial properties. So that's a good thing. So kind of related to the skin is also your epithelium, and we were kind of talking about this last day, and it's very similar to skin. It's usually not as many layers of cells, doesn't usually have that outer layer. Usually there's a lot of mucus secreting epitheliums, in your a lot of your systems, your digestive system, your reproductive tracts, and so on. And they have a lot of the same protective mechanisms as skin, and a little bit more because usually they're not as dry and they have better access to blood and things like that. So like I said, a lot of the secretions are the same, and then there's a few more things to add, like mucus. And we'll talk about mucus in a minute here, and these dendritic cells, which I want to talk about as well, which are really important for immunity and the mechanical defenses. So let's talk about some of these secretions that are found. And like I said, a lot of these things are in multiple body systems, right? Just underneath your skin, you can find lysozyme. You can find this in your tears. You can find it in your mucus secreting membranes. So what is lysozyme? Lysozyme was the very first enzyme ever discovered by scientists. So which is why it has a weird name. It doesn't have A's on the end. And it turns out that lysozyme actually chews up peptidoglycan. That's what it does. So if you take a look, here's some peptidoglycan found in the bacterial cell wall. Lysozyme comes in and it cuts holes. And it's found, like I said, in all sorts of places, your tears, it's found in eggs, it's found in just under your skin and in your blood, and it's antimicrobial. Lysozyme tends to be quite effective against gram-positives. Why is that? Yeah, they have way more peptidoglycan, right? And it's accessible to the enzyme. Remember, gram-negatives have the extra membrane? So lysozyme works really well, which is why a lot of eye infections we get tend to be gram-negatives, because they're more protected against the lysozyme that's found in your tears. Found in your nose, too, by the way. This is what Alexander Fleming was studying after discovering penicillin. He was studying this found in his nose. An interesting story. Okay, so let's talk about these dendritic cells. This is something that's going to have some overlapping concepts with innate and adaptive immunity. So these dendritic cells are technically a kind of a white blood cell, and they're like amoebas. They crawl around, and they've got these projections, right? That we would call pseudopods, right? And they're kind of funky looking anyway. And I kind of like to think of these as guard dogs, right? So you can imagine there's some sort of guard dog or security person, I guess, and they're just walking around. They're doing the rounds, and they're looking out for anything suspicious. And that's what these cells do. They move around like amoebas, and they move around through your tissues. Some of them have specialized names based on what part of the body they're found in. The ones that are found underneath the skin are called dendritic cells. And you can see they're floating around there. Like I said, they're in the lymph, they're in other tissues, and they're just looking for anything suspicious. And what they're going to do if they see anything suspicious, like let's say some bacteria, as they're going to grab onto it and bring it in by phagocytosis. And we're going to talk about phagocytosis a little bit later today, but that's where they grab onto something, engulf it, and bring it in, and often destroy it and digest it. I have a couple other pictures of them here. Here's another picture here of a dendritic cell. Just a little cute cartoon. Thought you might like that one. Here's kind of a little cartoon I found that sort of shows them, at a very basic level, some of the things they're involved in. One thing to keep in mind, I think I mentioned this last day, is that a lot of these immune cells, they don't just do one thing, right? And sometimes the way they do the things that they do, there's multiple ways to do it. So this is kind of just a general, what they often might do kind of thing, but not necessarily what they're always doing and how they do it. But there's a couple of important points here to mention. So you can see there's the dendritic cell. Like I said, it's walking around like a security guard or a guard dog or something like that. and there's something suspicious, so it pounces, right? In this case here, it's just some dead skin, but maybe there's some bacteria in there too. It's just, okay, whatever, we'll just grab onto it, brings it in by phagocytosis. There's some bacteria floating about, right? They get cleaned up too. And so you can see this is sort of trying to show what's going on inside the cell, right? And inside the cell, it's recognizing this isn't just, normal, something is suspicious here, right? It's bacterial in origin, okay? And so what it does is it sends out the alarm, right? So the alarm is usually chemical signals that might attract other immune cells or stimulate things like inflammation or stimulate other immune responses, of which there are many and several. And you can see this last part here is something we're kind of going to talk about a little bit. I'm not sure if we're going to get that today. Is sometimes they actually hold on to a piece of it, right? So that's why I like to think of them as a guard dog, right? You picture one of those comedy movies where there's the burglars coming in and the dog chases them and they make it out of the way, but the dog has a piece of their pants in its mouth, right? That's how I kind of picture them. They actually hold on to a piece, and they're going to hold on to it so that when other immune cells, kind of the adaptive response, they can actually sample it and take a look and see if it's anything suspicious, like, for example, maybe it was a viral infection you've had before. So there's a lot of interweaving between the innate and adaptive response. Kind of the main thing to know about dendritic cells, like I said, they're moving around looking for things suspicious. And if they find something that they think is not human, they're going to send out the alarm, which could lead to other immune responses. Okay, so that's kind of the important thing to know at this point. Okay, we will come back to dendritic cells. Okay, I know I have a picture from the textbook of a different one I was thinking of. There's all sorts of mechanical and physical kind of things we can do in our innate immune system. One really important system is this mucociliary system. So if you go way back, we were talking about cilia as part of a lot of protists. Remember, that's the kind of little finger-like swimming projections that are they're kind of like mini flagella. The human body has cilia, and they're not used for making our cells swim, but they are used for moving fluid, and they're all the way down our respiratory system. And this is called the mucociliary system because they have the cilia, which kind of flap around, and the mucus part. So what you're seeing here is what is actually happening to you all day long is we have mucus all the way down our respiratory system into our lungs, and the mucus is there to trap dust particles and potential pathogens that may come in while we breathe. And it moves the mucus up. The mucus goes all the way up to the back of your throat, and then you swallow it, and that's kind of what happens, right? And so it's clearing those passages. Usually we don't notice, it's small amounts. We're not noticing that swallowing part, except for when maybe you're sick, when suddenly you're producing way more mucus, and then you're sometimes coughing on it, or you notice the swallowing and it gets kind of gross. But that's kind of a normal thing, and like I said, it gets ramped up sometimes when we get sick, because the whole idea is to keep all those things out of there, right? Okay, so one more kind of mechanical defense I want to mention, partly because this is actually also a portal of entry, is the lacrimal apparatus. So the lacrimal apparatus is really just a system to basically continually flush your eyeballs, right? Your eyeball is almost a completely immune neutral part of your body in terms of there's no immune cells there. So we protect it by flushing it with salt and lysozyme and just continually flushing it. You can see what happens here. This lacrimal apparatus, it produces fluids, and the fluids kind of drain through the nasal cavity in the back of your sinuses, right? So that continual washing is hopefully keeping any pathogens out. But like I said, this is also a potential portal of entry, right? Alberta, for example, for me has been wonderful because whatever I was allergic to in Ontario, it's not here. And I didn't realize until I moved here how often I probably scratched my eyeballs. And I used to get colds so often because of just allergies and rubbing my eyeballs. So I'm introducing germs, particularly cold type viruses that are going to go and infect this part of my body, right? So it is a portal of entry, right? Washing your hands, again, is important so we don't, infect ourselves through this route. So like I said, there's all sorts of mechanical defenses. You can take a look. I think it's table 15.1 or something like that in the textbook that goes through a whole bunch of other possible things. We're not going to cover everything, just kind of touching on a few of these things that are part of the innate defenses. The last thing, of course, is do not forget our normal microbiome. Okay, these organisms, I showed you this exact slide before. These organisms are living on us and in us, and if we have the good ones, then they are able to at least inhibit, maybe not completely prevent, but in many cases that's what they're doing, is they're occupying niches that the pathogens would like to get a hold on. And so several mechanisms that we talked about before, just occupying niches, competing for nutrients, sometimes producing chemicals such as acids or proteins, bacteriocins, that can prevent growth of other organisms. And so that's also important to mention. And like I said, you know, we're starting to see in the scientific literature, and I don't think it's quite yet in the textbooks, where people are really talking about our microbiome as being almost an organ, a human organ, in terms of you know, having essential functions and those kind of things. And like I said, we're starting to see it sort of slowly creep into the textbooks. It's been in the literature for quite some time now. So that's kind of the first line of defense. Like I said, whirlwind tour here. You know, we've got the skin as kind of the big one, right? And all those secretions of the skin that are doing various things, whether it's on the surface of the skin or sometimes those things that are underneath, like dendritic cells and lysozyme and whatnot. Okay, and it doesn't seem like much, but this is actually, if you didn't have this part of your immune system, we'd be in serious trouble. And there's a lot of studies that have been done on animals in terms of removing these things one by one, like the ability to secrete salt and whatnot. And you know, it's actually does an amazing job of protecting us against a lot of invaders. All right, so second line of defense. We want to talk about blood and some of the things in the blood. Okay, and like I said, I'm going to skip complement and interferons. You can read about them. They're kind of interesting, but they're kind of technical. And like I said, I'm trying to make up for a little bit of lost time here and kind of just focus on some of the essentials. But let's talk about what's in the blood. So this is your blood. It's actually very complex. Everyone wants to think about those red blood cells. But if you think about what is in the blood, there's all sorts of things. There's cells and there's plasma. And the plasma, of course, is transporting nutrients and liquids to all of your bodies. It has proteins. Some of those proteins are involved in immune responses, and we're going to talk about some of those things like antibodies and other immune regulator type of proteins and so on. If we take a look at the cells, you can kind of divide them into three categories: the erythrocytes, which are also known as red blood cells, which are super important, but we're not going to talk about them today. Not important for the immune system. We also have platelets, which are important for blood clotting, and then the leukocytes, which are the white blood cells. So here's where it gets kind of technical with all the different types of cell types and those kind of things. And I know you cover some of them in anatomy and physiology, and I'll try to point out which ones are important for this class, because I don't know what you cover. There's at least 20 types, and sometimes even just depending on your sources, sometimes there's actually some of these things have multiple names, which I hate it when things have multiple names. I'm trying to remember one of them. So first thing to remember is that a leukocyte means a white blood cell, okay? So that's the first thing to remember, okay? Because those terms will be used interchangeably. All right, so if you take a look at the leukocytes, like I said, there's many different types. I found this, there's a similar thing in the textbook. I thought this one was just a little neater. So I thought I'd show this to you. So some of them end up, you can see they start off as stem cells, and we're kind of ignoring those ones over there that become red blood cells and platelets. And these are the ones we're focusing on. You can see there's a whole bunch of different types. And hey, look, there's our little dendritic cell, right? So I told you it was going to come back, and we're going to talk about it a little bit today. There's a few others here with adaptive immunity. We're going to talk about the B and T cells. And a bunch of these, you can see they have, like I said, some of these have different names. Some of them have different names based on the part of the body. And like I said, it kind of gets pretty complicated. A bunch of these are involved in the innate response. These ones are involved in the adaptive response. but they all have overlapping features, meaning that they all are involved really in both parts of the immune response ultimately. So like I said, immune system is unfortunately super complex, and we'd love to understand more of it. There's a lot we don't understand. Okay, so here's some of the leukocytes that you should know. I think I have some stars here. Right, okay. The stars, right? I didn't just put them there randomly. Those are the ones definitely you want to know, okay? And know a little bit about them. So first one are the lymphocytes. We're going to come back to those with adaptive immunity. That's the B cells and the T cells, right? Lymphocytes. So lymph, that's the lymph system, lymphatic system, okay? And cyte, by the way, means cell, right? So it just means it's a type of cell that lives in the lymph, right? And that's where your adaptive immune system is the most active. Macrophage, phage means eating, okay? So this one here is going to be involved in phagocytosis, which is cellular eating, okay? Definitely. Eosinophil, I don't really know where the name of that came from. This one is involved in allergies. And basophil is involved in inflammation and so on. And like I said, there's a lot more than just this. This is kind of just trying to, keep it to the amount of material that's appropriate for this course. Okay, so I told you we wanted to talk a little bit about phagocytosis. So this is the way that a number of immune cells will actually destroy those invaders by phagocytosis. So phagocytosis, phago means eating, cytosis means process of cell. So cellular eating, right, is what it is. The phagocyte is the cell that does it, and often they're called macrophages, macro meaning big, right? If you're going to eat another cell, it helps to have a little bit of size on you, and the dendritic cells, but they're not the only ones that do this. And they're very similar to amoeba. Like I said, they move around by changing the shape of their cytoplasm, and that's how they engulf things. So here's a phagocyte and eating a bacterium. I just want to show you a little bit in terms of the process of what is going on in terms of phagocytosis and how it works. So the first part of the process is that the phagocyte is often attracted to something, right? So again, if you think about the guard dog thing, they hear something, right? In the case of cells, they're not hearing things. Usually it's some sort of chemical. And so this could be a chemical released by one of those dendritic cells. Or in the case of sometimes, if our tissues get punctured, our tissues might release things like histamines. And those are chemicals that are attracting immune responses in immune cells, right? So this process is called chemotaxis. And you can see that little cell that's trying to crawl along and move towards whatever it is detecting, right? Then it grabs on, okay? And so you can see it's forming these little projections. I think I had mentioned them before. They're called pseudopods, right? So that means fake feet. And they surround the bacterium. So remember, we were talking about how sometimes if they have capsules or slime layers, this makes it harder for the immune cells to grab onto them, right? Because, of course, they're slippery and slimy. And then they get ingested into things that have many names. Sometimes people call these food vacuoles. You can see they're calling it a phagosome here. A zome means a body or a unit inside a cell. And so this is created by phagocytosis. And then eventually that phagosome or that food vacuole is going to fuse with the lysosome. So way back, I think it was lecture 2, we were talking about cell parts, right? We were talking about the nucleus and the ribosomes, and we're getting ready for the quiz. And one of the organelles I told you need to know is a lysosome. The lysosome is like a cellular stomach. It's actually an acidic organelle, and it contains digestive enzymes. And so it's digesting whatever it is that it found. So in this case, it looks like a little bacterium there. So a lot of it is going to get destroyed, not always. And depending on the type of phagocyte, this last part of the process kind of depends on what's going on. You can see it has this note here that neutrophils release debris. So why are they releasing debris? Because they want to have the other parts of the immune system come and just take a look. What is it that you found there, right? Is it something we should be concerned about, kind of thing? In some cases, this is a bad thing, right? We talked about those endotoxins last day. Remember the LPS, the lipopolysaccharides and gram-negatives? They get into your blood. It can cause the immune system to get a little overly excited. So sometimes it's a bad thing. Other times it's a good thing. Sometimes in the case of dendritic cells, they don't spit it out. They actually have mechanisms to hold on to it. And it's presented is the word we use. It's actually held onto at the cell membrane. So like I said, there's a bunch of different kind of scenarios that might happen here depending on the cell. It doesn't matter exactly that you remember which cell does which, but that this part of the process may stimulate further immune responses, either by secretion or holding on, and that's called antigen presentation. That's what the term is there. Okay, quick little video here for you. So happy to find this. This is just showing a little bit of phagocytosis, and if you kind of watch what's going on, all those little things are bacteria. I'm not sure if this is sped up or not. I have no idea, but it's trying to show some phagocytosis happening in these immune cells. Like I said, I kind of think of them as guard dogs that are chasing down those burglars or invaders, and in this animation, they're doing it very, very quickly. And then some of these, this is not animation, that one was an actual video. And there it is, releasing the proteins, and that might be attracting further immune cells. All right, that's kind of what I wanted to show you. The video is a little busier than I want it to be, but kind of short and sweet. There's some pictures of some phagocytosis from the textbook. You can see it's pretty easy to imagine a white blood cell engulfing a bacterial organism, but in some cases they are actually engulfing cells that are almost their size, right? So you can see a yeast cell as a eukaryotic cell, so it's going to be bigger than a bacterial cell, and it can actually be destroyed by phagocytosis. Now, a yeast cell is not a big eukaryotic cell. So you can start to picture that there are larger invaders, right? Particularly if you start to think about the worms. And the immune system doesn't just kill invaders. Sometimes it will actually kill cancerous tumor cells, so abnormal human cells. Sometimes it actually will destroy our own cells that have been infected by a virus. So think about it, if your own cell is infected by a virus, that's bad. That cell needs to be destroyed. And it could be a very large cell. So we have to have other ways of killing, right? And so we have phagocytosis, and oh, sorry, getting ahead of myself. I thought this slide was earlier. This is showing some of the phagocytes around the body. So there's the dendritic cells, right? Found in the, like I said, just underneath the skin. And there's a whole bunch of these. Like I said, they often have different names in the different body systems, like these ones here. Kupffer cells, Kupffer cells, I guess. Remember learning about those at some point a long time ago. Probably named after a guy named Kupffer. I can't even say that. Looks like it's easy to say, but try saying it fast. Anyway, like I said, there's a whole bunch of these in the body. We don't need to learn all of them. Just learn the ones I had the star beside. Okay, like I said, getting ahead of myself. Can't just kill things by phagocytosis. Sometimes we have to kill things by non-phagocytic killing, which is kind of a different story, right? So like I said, picture these large organisms, worms, tumors, virally infected cells that are too big to kill by phagocytosis, right? So this is called cellular mediated killing. There's actually a bunch of names for it, and cytotoxic killing. So cytotoxic, right? Cyto is cell, toxic is death, I guess, right? And there's a few different mechanisms in how this works. And I kind of like to think of it as the kiss of death, right? So picture some sort of movie, and you have, let's say it's a female assassin and she has poison in her lipstick, right? Or something like that. Same idea, right? So what happens is the the killing cell, the immune cell, of which they have many names. Some of them are called killer cells or natural killer cells. They go and they inject a toxin into that target cell. And that's actually what's being shown here in this little cartoon. You see these little granules in there. They're toxins, and they're going to do something to that cell. And there's many mechanisms. Sometimes they just punch holes into the membranes. Sometimes there's other mechanisms for killing. And like I said, I don't want to get into all of this. Sometimes what the cell does is basically tell the other cell to commit suicide. Like I said, there's a few ways of doing this. And I don't want to get into all the details of them. Some of them are called natural killer cells because they can do it automatically. Other cells need to be activated to become killer cells, right? So you can imagine all sorts of, I'm sure, assassin movie type analogies here. Sleeper agents and things like that. So brings us to these guys here, these eosinophils. So these eosinophils are involved in killing worms, okay? And you can see these granules, they're just loaded with these toxins. And that's great, right? We want to protect our bodies against parasitic worms and other infections like that. The problem is that in modern, developed, wealthy countries such as Canada, we are not getting worm infections very regularly. And so what do we do with these cells, right? So these cells get a little restless. And so it turns out that they're also involved in allergic responses. And so we're going to talk about allergic responses and that relationship with this part of the immune system in part 3 of this lecture. But there is a relationship there, right? So generally, if you get infected with parasitic worms a lot, you don't get allergies. If you live in a place of the world where you're not getting affected by worms very much, then you have a greater chance of developing allergic diseases. And that's the same part of the immune system, unfortunately. So a bit more on that. We'll touch on that a little bit in part 13-3. So there's some of those eosinophils, and they're attacking a fluke. And you can see they're really attacking it. And they're going to be, like I said, basically secreting those toxin granules. And in this case, probably punching a bunch of holes into the membranes of the fluke cells and just killing it. Okay, so we're just talking about all these cells, and they're found in the blood. All right, so we have sort of those two mechanisms, phagocytosis and cytotoxic killing. And some of these things are done automatically. There are certain chemical markers that make it obvious to your immune system that this is a bacteria or this is a worm or something like that. And like I said, some of this will overlap and come back in the adaptive part of the immune system. There are a bunch of other things that are found in the blood that are sort of fall in this category as well. Like I said, part 2, second line of defense is the blood. It also has lysozyme, also has defensins. We also have this complement system and the interferons, which I'm going to skip. You can see there's sort of some definitions here. Interferons, I think I mentioned them way back in, I can't remember which topic, somewhere at the beginning when we were talking about viruses. I mentioned that's one difference between viruses is that they're sensitive to interferons. And there's a whole bunch of different ones. I think there's a big table in the textbook. Some of them are designed for attacking double-stranded RNA, for example. Right? Double-stranded RNA, where do you find that? Viruses. Some viruses have double-stranded RNA. So that's what interferons do. A whole bunch of different mechanisms. You may have heard of the word interferon if you've ever known anyone going through some sort of cancer therapy. That's one of the cancer therapies is they are getting injections or IV of interferons. And what that is doing is trying to ramp up the immune system to deal with basically ramp up the immune system is what they're trying to do, to make a long story short. The complement system is a bunch of proteins, and they're kind of like, just trying to think of a good analogy for this, but they're little warning bells, right? So it's kind of like the bank robber, the button, right? They show in movies again, right? And then what does that button do? You press that button and it alerts the police and the fire truck and you know, the FBI and everything like that. And so that's kind of what the complement system is. It's like these proteins, and they're going to activate a whole bunch of signals that may lead to other immune responses. But I'm not going to get into them any more than that. I may have some slides I was going to skip. Yeah, I'm going to just kind of skip these slides. But there's the complement system, like a cascade. You know, this leads to this, leads to this, leads to this. Okay, there's the complement system causing an immune response and punching holes in these cells. And there's interferons. Okay, last couple parts of the innate immune response I want to talk about are inflammation and fever. Okay, so let's talk about what those things are. Inflammation is a general, non-specific response to some sort of tissue damage. Okay, so you could imagine that that could be, you know, let's say an insect bite, or you get scratched by a thorn, or whatever, right? Or it could be something internal. Sometimes people get a viral infection, and you know, people with, let's say, pneumonia or COVID-19 or something like that, they get a lung infection and they get inflammation down their chest, and that can lead to extra fluid in the chest and coughing and so on, right? So it can be chronic or systemic. There's all sorts of different types of inflammation. But generally what it is just a general reaction to tissue damage of some sort. Short-term inflammation is actually a good thing. And I'll explain why that is in a minute in terms of, you know, actually helping you to heal and recruit other parts of the immune response. Chronic inflammation is unfortunate, where it can lead to sometimes collateral damage to body systems. Could be arthritis, could be some sort of severe symptom, all sorts of other things, right? So let's talk about what's going on with inflammation, okay? So an inflammatory response is really just designed to basically open up these blood vessels in that area. So you can see here's kind of your normal blood vessels. And what we want to do is kind of open these up, right? And you want to bring in more blood. So you want to bring in all, you know, the nutrients and the oxygen so you can heal. You want to bring in platelets so you can clot, right? Like if you've got a scratch and it's bleeding, you want to clot right away. You also want to bring in those immune cells. So you're kind of just opening things up and trying to accelerate the defense and healing process. So what does that lead to? It leads to a little bit of red in the area, sometimes a little bit of soreness, sometimes a little bit of heat. Sometimes it kind of gets itchy, right? It's kind of in the short, small scale sense of everything that's going on. I think I have another picture here showing there's kind of the cracks in the tissues open up, and you've got these phagocytes and dendritic cells are kind of getting in there, and they're, like I said, they're you know, think about it. You scratch yourself on a thorn, you could be introducing bacteria into that wound, and so you want to bring in, you know, bring in all the defenses and deal with what's going on there. Oh, I thought there was a different slide here first. By the way, this is just kind of a cute little, what's this called? Not, I guess, acronym for thinking about the symptoms of inflammation: sharp, right? Swelling, heat, redness, pain, and pus, right? So, bet you guys see a lot of those. You see a lot of those in some of your classes? Yeah, there's sort of memory mechanisms for, there's lots of memory mechanisms for kind of learning things. I had one student a few years ago, and she used to do that on all her tests, and there'd be these weird sentences written down, right? And there's one time there's something about how all the Fort Montgomery police were into crack cocaine, and I'm just like, okay, you got to explain this one to me. And she's like, oh, it's what do they call them? A pneumatic memory thing, and I think it has something to do with the citric acid cycle, right? In terms of, you know, each word represented another word. Anyway, she had some great ones in there. That was one of her study mechanisms. This was the picture I thought was first. So you can kind of see everything that's happening here. You got the puncture, right? And the mechanism behind the puncture, in terms of recruiting all these blood cells and opening up those vessels, is histamine. Okay, so that histamine is a chemical messenger, and it does all of those things. It you know, opens up the capillaries. It you know, brings in the extra blood and nutrients, causes the heat, and unfortunately some of the itchiness. And anyone who's ever had to deal with allergies knows what histamines are, and we want to block those things because sometimes they're causing all of those uncomfortable allergy symptoms that are making our life unpleasant. But that's kind of the inflammatory response in a very general sense. Okay, last part of the innate immune response I want to talk about is fever. And I will say this right off the top, is that there's a lot of professional disagreement on how fever should be necessarily handled in terms of those kind of things. It's interesting to see the shift of these things over the years, because now there's a lot more healthcare providers that are saying, no, if you have a mild fever, you should just let it go. That's fine. It's actually part of your immune response. So what is a fever? A fever is a response in your body, usually by the hypothalamus. The hypothalamus is part of your brain that regulates temperature, and it's detecting something, and so these are called pyrogens, so pyro, fire, right? And that are saying, okay, we should turn the heat up a little bit, right? And there's all sorts of reasons why people might have fevers. Generally, particularly due to infection, there's parts of bacteria and viruses that tell your body to do so. Sometimes interferons tell your body to run a fever, and so on, right? And so the whole idea here is that if you know the basics about chemistry, warmer temperatures speed things up. So this is going to enhance a lot of the immune response, even though it sucks, right? It's uncomfortable, you're sweating in your bed or whatever. It does enhance the immune response. And in the case of some pathogens, it actually makes it harder for them to actually live. Particularly a lot of parasitic worms, they barely like 37 degrees, never mind 38, 39 degrees. It makes it very uncomfortable for them to live. Thought I had another slide over here. There we go. How does it help? It enhances interferons, inhibits growth, enhances macrophages, enhances tissue repair, right? So, and one of the problems with fever, sometimes too much can be bad for you, right? So in terms of, exactly what temperature we should be trying to control a fever is some of the debate in terms of, and that will of course depend on someone's age. Fever in children versus fever in adults is a bit of a different thing too as well. And so that's something, like I said, in the years I've been observing, talking to different physicians. Sometimes, there's different philosophies on when and how to manage a fever. So I'm not going to comment on that anymore. Okay, so that was a whirlwind tour through innate immunity. Okay, like I said, it's kind of just a big long list of here are some things that protect us, things that are non-specific, relatively fast mechanisms for protecting us, and you're going to see some overlapping concepts with adaptive immunity. So let's get into adaptive immunity. Any questions at this point? All right, so take a look at your notes. There's a bit more information. The textbook goes into this in an excruciating amount of detail. There's a lot of things that we didn't cover, and I will not test you on those, okay? I will just what we've discussed here today. Okay, so this lecture here, I'm guessing will probably not finish this for sure. There's quite a bit of kind of stuff to cover here in terms of, I'm at the point where I'm wondering whether I should be breaking it down into two topics, one on vaccines and one on adaptive immunity, but I still have it all grouped together as one. This is where it gets super complex, okay? And like I said, I'm going to try to, just cover a certain amount of basic stuff, and sometimes the textbook gets a little bogged down in the details on this particular area. But let's take a look at this. So there's our slide I showed you before. So one thing to note about adaptive immunity is it's much more specific and, unfortunately, a little bit slower. but it's very, very good. Without adaptive immunity, we would be in huge trouble. We'll talk a little bit about immune disorders in part 3 as well. Okay, so I want to talk about some features of adaptive immunity in terms of trying to understand what exactly it is doing, okay? So feature 1 is specific, and I mentioned that just a moment ago. And what does that mean? It means it's recognizing a very specific antigen or molecule. So specific that we're talking about not necessarily recognizing a virus, but actually recognizing maybe just one glycoprotein on that virus. That's how specific it can be. We can recognize toxins, we can recognize all sorts of things. Number 2, it's inducible. So what does that mean? It means that, it's kind of dangerous to have an immune system in overdrive all the time. So we kind of tone it down when it's not important. And so inducible means we can turn it on and activate it when we encounter whatever it is that we're trying to deal with. Clonality. So clonality and memory are kind of combined here, but basically, we have, if you think about it, right, how many viruses are out there on the planet? We don't know the number. We don't know the answer to that. Probably billions of different viruses, okay? And how many immune cells do you have? Also billions. But we're not going to have all of those immune cells with many copies and all over the place. It's going to be too much. So you just have a few immune cells, but they have the ability to clone themselves. That means copy of themselves when the need arises. And that's what clonality is about. No response to self. Hopefully that one's obvious. What we're trying to avoid is having an immune response against our own tissues. That would be an autoimmune disease. We don't want that. It happens sometimes because this system is super complex, and so sometimes it doesn't always work properly. Again, we'll talk about autoimmune diseases next unit. And then memory, we can remember previous infections, right? And this is important as well. And this is actually the whole basis behind like vaccines, right? So that we don't have to have that previous infection, have a memory of what to destroy when we encounter it for real. Okay, so just some things to think about as we go through the adaptive immune response in terms of how these things are carried out, because these are essential features of adaptive immunity in terms of how it functions. Okay, so other part to think about before we talk about how it works is the players in the system here. Okay, they are numerous, and we're not going to cover them all. First one is the lymphatic system, and we covered this in anatomy and physiology. Yeah, this is something that's kind of hard to explain to people if they've never heard about the lymphatic system. It's kind of like a circulatory system, another circulatory system, right? You have your main circulatory system with the blood and the hearts and and all your capillaries and veins and arteries and all those kind of things. This has its own little lymphatic vessels, and it pushes lymph through, but there's no heart that pushes through. So this is a much slower kind of system. And so what is this lymph stuff, right? It includes liquids and plasma and waste from the blood, right? And so you think about your cells are metabolizing all the time. There's wastes and there's, all sorts of things. So this is kind of trying to deal with those things. And this is actually a good sampling ground for your immune system to see what's going on in the body. And so there's lymphatic systems all over the place. There's a whole bunch of different parts to it. You can see, like, for example, there's lymph nodes. These are larger areas that, where the immune cells gather, and sometimes they swell up when we get infections. And they're kind of all over the body. Some of the places that are more obvious are under your armpits or in your neck, but they are found all over your body. I can't even remember how many there are, quite a few. There's also other tissues that are important to mention, such as the bone marrow. That's where your blood cells are, that's where the stem cells for your blood cells are. So that's where they're made. The thymus as well is important for the T lymphocytes, which we're going to talk about here in a bit here today. All right, so the lymphatic system is kind of like the battleground. This is where the immune cells are found and where a lot of the immune response is taking place. Of course, we also have the villains, which are the antigens. Okay, antigens, by the way, in the textbook and all over the place is abbreviated as AG. Okay, so you'll probably see that on some of my slides. This includes all sorts of things, right? Usually when we say antigen, we don't mean an entire organism. We usually mean one molecule. So maybe a surface glycoprotein found on a bacteria, or a carbohydrate found on a capsule, or a spike protein found on a virus. And in some cases, it's not even attached to an organism. An antigen can be a toxin. Sometimes some of us might be allergic to things like penicillin, and that's a molecule. So an antigen can be a relatively small molecule in some cases. Usually, though, we're talking about some sort of surface feature on an organism of some sort. Okay. So these are the villains, the invaders, however you want to think of them. These are the things that we're trying to usually destroy and get rid of. And then there's the antibodies. So we talked about antibodies a little bit. Told you we're going to come back to them. Here on the left is an image of an antibody. They're proteins, and they're actually complex proteins in that they're made out of one antibody is actually 4 proteins. 2 big ones and 2 small ones. They're called the heavy chains and the light chains. I would not have named them that way. And you can see this is trying to show how they're connected together. So you got one heavy and one light, one heavy and one light, and they form kind of this Y shape, which you can see on the left. The molecules do look like kind of Y shapes, right? So a couple of things to point out what is going on these antibodies. Is there an antigen? There is an antigen binding site. And actually, the antibodies have 2, right? They're Y-shaped. They've got 2 arms. And that's found over here in the top. You can see I have it labeled in the top right. Sometimes that's called the variable region. And why do we call it the variable region? Because that's the part that the immune system can mix and match and switch up, because, of course, there's many possible antigens, right? You could have an antibody for the glycoprotein found on the varicella virus. You could have a glycoprotein found on the carbohydrate found on pneumococcal pneumonia, and so on, right? So many possibilities. That's why it's variable. The other part of the antibody is down here. This is called the constant region, and so that's kind of just you can think of that as an adapter that's recognized by immune cells. Okay, and there's only a few different parts like that, not like a billion. Like the variable region, there's probably billions of possibilities, whereas the constant region, there's only really a few, and that's going to be recognized by immune cells. Okay, so we'll talk about how these things work here. I think I have that right here coming up. There's a couple of pictures of antibodies binding antigens, and you can see I like this picture here because you can see that what they're trying to show is that there's different possibilities all in one bacterial cell of different antigens. And you can see some attaching to the surface there, attaching to probably some carbohydrates and so on. And this picture here is showing that there's 3 different antibodies actually recognizing different parts of one molecule. So I want to talk about how antibodies do what they do, because this is the huge part of the arsenal of the adaptive immune system. And they do a whole bunch of things. I want to talk about each of these kind of one at a time. The main things they do are what we just talked about a few minutes ago, is they lead to the killing of the invaders by either one of those 2 mechanisms, right? Either by phagocytosis or by the cytotoxic killing, right? So that's kind of the main way we kill things. And there's different routes to getting there. So let's kind of talk about this. The first one is called opsonization. I don't know why we have to have a fancy word for this, but that basically means the process that leads to phagocytosis, right? So you can see there's our little bacterium, or it could be a viral particle, could be a toxin, doesn't really matter. It's recognized by an antibody. And then that constant region is recognized by a phagocyte. So this basically tags this thing. And there's a little tag that says, hey, come and eat this, okay? So the macrophage, the phagocyte comes along, grabs it, and brings it in by phagocytosis. Bacteria destroyed. Check, gone. So this is good. We're trying to get rid of the invaders. So opsonization, unfortunately, another new word. Like I said, it basically means it's leading to or stimulating phagocytosis. All right, number 2, antibody dependent cellular cytotoxicity, okay? One of these things where some scientists decided they wanted a big long word and acronym for this. It's very descriptive. If you take a look at what's going on, it's basically stimulating cellular toxicity, right? That kiss of death thing that I was telling you guys about. You can see NK actually stands for natural killer. It doesn't have to be a bacteria. This could be a much larger cell, but basically it's tagging the thing, and the natural killer cell comes along. The natural killer cell is not a phagocyte. It can't do phagocytosis, right? It doesn't have that mechanism to be able to do so. But what can it do? It can kill things by cytotoxic killing, right? Which is injecting toxins. You can see what they're trying to show in this picture is this perforin is a thing that goes in the membrane and basically leads to big pores, and that bacterial cell or whatever that is that's being attacked is going to be destroyed. Okay, so those are the two functions. Like I said, there's other routes to get there, and that's what the other three functions of antibodies are. So function 3 is activating the complement system. I didn't want to talk too much about the complement system, but other than the complement system is like a messenger that leads to other immune responses, which usually leads to phagocytosis or cytotoxic killing. So I'm just going to keep moving on that. So what else can antibodies do? They have two arms, so they can help to clump things up. We've already talked about this process of agglutination as being used in diagnostic techniques. I showed you some pictures of agglutination processes, and you can do this to detect your blood types, so they clump things up. So what is this doing? Well, this is helping to make the pathogen more visible to the immune system, right? Larger things are easier to see. So this will often lead to enhanced phagocytosis, right? So clumping things up leads to enhanced phagocytosis. That's kind of the main thing that's going on here. This is kind of reviewing. Remember, we were talking about using the agglutination process to detect your blood type. So if you take a look, here's the 4 blood types, and you've got different antigens based on what your blood type is. So if you have A-type blood, you have A-type antigen. If you have B-type blood, you have B-type antigen. Those are carbohydrates, by the way. If you have AB, you have both. If you have O, you have none. And you can test somebody's blood by looking for agglutination to see which antigen they have. So if you take a look, if you use the anti-A antibodies, anti-A means it's binding to the A antigen, and so the person with the A-type blood would clump, and the B-type, but not clump with the B-type antibodies, right? Opposite occurs if you have the B-type blood. You have AB-type blood, you get clumping in both. If you have O-type blood, there's no clumping. So just a diagnostic use for antibodies, just kind of a quick review of something we talked about before. Okay, what else can antibodies do? They can neutralize things. So what do I mean by neutralize? It means that, think about it, you have a virus, the virus particle is coming along and wants to bind to your cell, but if you can block it, then it's useless. And so that's actually what antibodies can do. So you can see in particular this case here, this toxin. So we have, most of us when we're children, we get vaccinated against diphtheria and pertussis toxins. So the diphtheria and pertussis vaccines do not actually have the organism. They are actually, you're getting vaccinated against the toxin. So many of us don't even know it right now, probably have diphtheria. But the organism is not causing us to be sick because the toxins are bound immediately by the antibodies, and they're neutralized and not able to cause any illness. I can't remember what the stats are. I think it was 20% or something like that of people are harboring diphtheria, right? And it's become tamed, right? We've tamed the organism. But it can do the same thing with viruses. We can block viruses, we can block bacteria. Usually this, again, often will lead to phagocytosis as well. Okay, so like I said, really, what are antibodies doing? They're labeling things and often marking them for phagocytosis or cytotoxic killing. You know, the bonus is sometimes we're neutralizing them as well in terms of what antibodies are actually doing. Okay, so We have the lymphatic system, the battleground. We have the antigens. These are the targets. We have the antibodies, which are the arsenal of our immune system. And we also want to talk about cells and food. By the way, you may have noticed, let me just go back a few slides. Way too many slides. Back. Notice over here, antibodies are abbreviated AB. They're also called immunoglobulins, right? So globulin basically means protein, okay? A certain type of protein. Immuno means your immune system. So sometimes antibodies are called AB, sometimes they're called IG. So fun, multiple names for things, right? We just didn't want to make the immune system any more complicated. We do that anyway. So one thing I wanted to say is that there are different types of antibodies. I won't test you on this, but you might see in your travels references to IgA or IgM or IgG or something like that. And they all have, slight differences in structures. You can see IgG, for example, is your classical Y-shaped antibody. IgA, sometimes you have a couple of them kind of strapped together. IgM, you actually have 5 strapped together, right? So extra arms. And so these IgM antibodies can actually be quite effective in neutralization quite a bit. And there's IgE and IgD. So what are some of the differences? In terms of what is going on with the immune system, they all have slightly different roles and overlapping roles as well. You can see that IgG, one of the essential things about IgG is it's small, and this can actually cross the placenta. So this is great, right? As babies, you probably don't realize, but this is awesome because there's actually a lot of protection that you get from your mother against all sorts of diseases that have crossed the placenta. And that gives you immune protection for several months, even after birth. IgA is also involved in some of that same mother-to-child protection and can actually be secreted through the breast milk and survives digestion in the body. IgM and IgE, you can see, like I said, overlapping kind of protections in terms of what is going on. I can't remember. I think IgM is one of the first ones to be secreted. I have to look that one up. And like I said, with all those multiple arms, it kind of it helps its efficiency and whatnot. IgE is involved in attacking parasitic worms and allergies, unfortunately. And IgD, apparently we don't know what it does yet in terms of exactly how it's in our immune system. Still working on that one, I guess. So don't forget, we did talk about a bunch of serological tests. One of them was the ELISA test, and that's shown here. You can see all these pictures are showing the antibodies. And the whole idea behind an ELISA test is to recognize some sort of antigen. And that antigen could be a pathogen part, or it could be one of your human antibodies, right? So in the case of HIV, for example, the amount of viral particles in somebody's blood is so small, it's super hard to detect. So when the HIV test, you're actually looking for human antibodies against the HIV virus because they're found in a higher concentration, much easier to test. I'm not going to go through all the steps of this. Just a reminder that these rapid tests are basically similar technology in terms of what is going on. And there's an example of a rapid HIV test. All right, so part D are the cells. And so we want to talk about these lymphocytes, right? So lymphocytes are a type of leukocyte. Remember, leukocyte is a white blood cell. Lymphocyte, we're specifically talking about the B cells and the T cells. So let's talk about those guys. There's a picture, by the way. You can see some blood, and it's just showing the different types. Usually I actually have some blood cell models I was going to pull out, but they were put in storage because of some renovations, so I wasn't able to access them. I was going to bring them to class and show you them, but so picture is okay, I guess. It's not as nice. Okay, so these are called B cells and T cells, or B lymphocytes and T lymphocytes. Okay, those terms are used interchangeably. So what is the difference between these things? They're all made in the bone marrow. That's where your stem cells are. But all of these things, these cells have they have antibodies on them, and these antibodies are going to have the potential to recognize all sorts of antigens. And there's sort of a genetic random thing going on here in terms of what it's making. And so this antigen binding site could potentially recognize anything. And so what we don't want it to do is to recognize human tissue. So we have a process that we call maturation or clonal deletion. And what does that mean? It means we eliminate the cells that are potentially dangerous. And so that's done for the B cells in the bone marrow. So B for bone marrow, right? So that's why they're called B cells or B lymphocytes. And the T cells, it's done in the thymus. So the thymus is a little organ that's found somewhere up here. I think it's just below your collarbone, if I remember correctly. You know, it's just a little tiny organelle, and I think that's its only function. I'm not entirely sure, to tell you the truth. But that's where the T cells are matured. And like I said, they eliminate the ones that are potentially self-reactive, which is a really important part of our immune system. There's that the terminology clonal deletion, right? That means you're killing the clones that are bad, right? So these T cells and B cells are sometimes called clones, right? Which is kind of a funny word because a clone usually means an identical copy, but it turns out when the immune system clones things, things aren't always identical because they're trying to have some randomized stuff going on so that we can recognize lots of invaders. But it basically means you're killing the bad cells, right? So you can see here's a little diagram. You can see we've got a whole bunch of different potential B cells or T cells being made. And the body is like, okay, wait a second, there's some here that recognize self. So those ones are going to be killed or deleted. So clonal deletion is just getting rid of the ones that could be self-reactive. So there's another term that's important to talk about for these immune cells. It's called clonal selection. I'm going to show you a really good video here in a couple of minutes, if we have time, that is going to show this process happening. So the other thing we want to do, besides get rid of the bad ones, is we want to make extra copies of the good ones. And so this is called clonal selection. So you can see in this case here, you've got this little lymphocyte, and it's just moving around the lymph system, and then maybe it finds something, right? So maybe now you got an infection of some sort, or it's a vaccine or something, and it binds it. And so that is a signal for this cell to say, okay, you are needed here. Make copies of yourself and be ready. And so these ones here get copied, and some of them carry out the immune response, and some of them become memory cells. So that's going to be part of the immune response we're going to talk about. And memory cells are long-lived cells that can be reactivated at future days to combat that antigen. All right, so let's see where we are here. So there's a whole bunch of different types of these cells, by the way, particularly when you get to the T cells. I can't even remember how many different types of T cells there are. It gets super complicated, and I don't want to go into all of that. Here's a few, right? Some of the things that we've kind of talked about when we talk about some of these T cells are cytotoxic, so they don't do phagocytosis, but they kill things by cytotoxic killing. Some of them are naturally cytotoxic, so they're called natural killers. A whole bunch of these T cells are regulatory cells, and so B cells sometimes need to be activated by T cells, and T cells sometimes interact with B cells in order to carry out that immune response. And it's like this crazy, really complex committee, right? Except for it's probably a lot more efficient than any committee on the planet because it works really well. So there's a lot that could be discussed here that we're not going to. The main thing to say is that there are lots of different types of these cells. We're just going to mostly distinguish between the B lymphocytes and the T lymphocytes. Okay, the last category, which we're also kind of just going to glaze over, is that there's all sorts of chemical signals and mediators out there that we could talk about that we're not going to. These are chemical messengers, and they go by a bunch of names. One of them are the cytokines, and I know they've been talking about cytokine storms along with COVID-19, and I actually have a slide for that, so we'll get to that, what that is in a minute. And these are involved in basically communication between these cells. So I mentioned, for example, the dendritic cells send out a signal. And that signal is often a cytokine, and that cytokine is an attractant to bring along other macrophages, another part of the immune system. And there's a whole bunch of these, and some of these are involved in therapies. You can see I have some list here, interleukins and TNF-alpha. Can't remember what that stands for. But these are sometimes used in various therapies, chemotherapy and things like that as well, to sort of try to enhance the immune system as well. I was going to mention cytokine storm because it kind of has been coming up with COVID-19 as something that happens to some people. Some people get really bad, really fast, kind of in that second week. And this is believed to have something to do with these cytokine storms. So what is going on between a cytokine storm? So the cytokine is an immune chemical that enhances and brings along other parts of the immune system, right? So it's normal and natural. But sometimes people end up with kind of an elevated response. So too many cytokines end up kind of just coming out into the system. And it's kind of like this, right? It's kind of like a runaway train. So things are just getting out of control, and how do you stop it now? And that's what happens. In this case, in the case of some of these COVID-19 patients, people's lungs are getting flooded with liquid, and they're in some cases like they're drowning, is what's happening. In other cases, you're looking at kind of inflammation that's happening all sorts of crazy parts of the body, and can be and cause all sorts of trouble. So don't worry about the chemical signals. We're not going to be talking about them anymore. Just know that that is kind of how they communicate with one another. Okay, so I think just enough time to kind of overview these things here. Just trying to think. Probably have just enough time to show this video as well. So I think that's probably where we're going to end up today, and we'll come back and kind of review this next day. So you can think of the adaptive immune system as having two kind of systems: the B cell system and the T cell system. So we call this the humoral response and the cell-mediated response. Okay. Again, you know, more words, and why are we using humoral? I don't know. Humoral is an old historical term for blood. Why don't we use blood? I don't know. Cell-mediated, that means, you know, not usually phagocytosis, I guess. Again, it's a little bit confusing. So, but here's the thing. We have differences between these B cells and these T cells, right? The B cells, generally what they do when they find an antigen is they secrete soluble antibodies. And then the antibodies do their thing, right? Remember all those things we're talking about, what antibodies do? They do phagocytosis. They recruit other immune cells for cytotoxic killing. Sometimes they neutralize viruses. Sometimes they activate the complement system and so on. T cells are more of the natural killer type, right? They do that kiss of death stuff, and that's how they kill things. So you're looking at kind of two prongs of the immune system. The B cells are usually targeting smaller things that can, in fact, be killed by phagocytosis, or at least many of them are. The T cells are often targeting, you know, sometimes bigger things or cells that are already infected that we need to clear out. So what I want to do is I want to show you this video here. It's a very good video. I think it was made in New Zealand, so the guy has kind of a cool accent, and he's going to talk about the humoral response. It's a very good video. Yeah. Yeah, antigen. Yeah, like the B cell, the B cells are secreting antibodies that bind antigens. Yeah. Like I said, the problem with this is the language. A lot of these things have so many different names, and trying to be consistent with our textbook and at least the main way that people call these. I've not heard it called that, but it makes sense. Yeah, good question. So I'm going to play this video for you. That might be all we do, but it's a really good video on the humoral response, and it reviews a lot of the concepts that I just talked about.
In 1960, Sir Frank Macfarlane Burnet was awarded the Nobel Prize for his contribution to the field of immunology. Burnet's clonal selection theory revolutionized our understanding of the immune system. To this day, Burnet's research remains the foundation of the way we think about disease and how the body responds to it. Clonal selection is the central mechanism by which your body fights infection. The following visualization presents clonal selection during an infection by strep throat bacteria. This patient has a strep bacterial infection causing a red, sore, pussy throat. The tiny round strep bacteria thrive on throat tissue. The bacteria are coated with hairs called M protein. M protein is what your body recognizes to be foreign. In response, your body will launch an immune attack against it. Your lymphatic network is an important part of the immune system, detecting any infections that arise in your body. The M protein hairs drain into the lymphatic system around this patient's neck. The lymph node is crammed full of B cells. B cells are guardians of your immune system, scanning the body for foreign material. Arriving at the lymph node are M protein hairs from the bacteria. Your body contains billions of B cells. Each B cell is unique and is sensitive to only one kind of foreign infection. By chance, one B cell is sensitive to the M protein hairs from the bacteria. This is the molecular surface of that B cell, magnified 10 million times. Its surface is covered with receptors that recognize M protein. The receptors grab onto the M protein hairs. This triggers an activation signal to travel into the cell. The activated B cell migrates and begins to replicate. In a few days, thousands of copies of the selected cell are made. This is the central idea in Burnett's clonal selection theory, where one cell is selected out of billions of B cells and identical clones have been created from it. Therefore, the cell is clonally selected. This creates an army of B cells that mass produce M protein antibodies and release them into the surrounding fluid. The antibodies circulate in the blood and travel into tissues. They bind to the M protein hairs on the strep bacteria. This flags them for destruction by the immune system. Other immune cells ingest the antibody-covered bacteria. They destroy the bacteria and eliminate the infection.
Okay, so that's it. Cool video. Kind of review some of these. We'll come back and I'll kind of review some of these concepts in terms of how the humoral and cell-mediated response work, and kind of cover some other things next day. So thanks for coming. We'll see you next day.